Thanks. Thanks, Sophie. So I'll just share my screen. Is it okay? Can you see it?
So it's
okay.
Yes, now it's in the presentation mode.
Yeah, good afternoon, everyone. And again, thank you for that introduction, Sophie. And yeah, I'd like to thank the organizers of this seminar series, Amy, Aidan, and Sophie, for inviting me to give this talk. And to all of you who are here, thanks for your time to attend this seminar. And what I will be presenting today is part of our ongoing research on the last 7 million year history of the southern sector of the Indian Ocean with collaborators from the University of Salamanca and Cardiff University, and some members of the IODP Expedition 361 Science Party. So before I start with the presentation, let me just turn off my video. Yeah. So yeah, for significant changes in the Earth's climate system occurred over the last 5 million years. And this makes this time interval the focus of numerous studies over the past several decades. And always of particular interest, and is very popular, is the onset of the Northern Hemisphere glaciation at around 3 million years, the orange box over here. And this has been known to set the Earth's climate into entering the bipolar, orbitally driven 141 kiloyear glacial interglacial cycles. And these cyclicities change from 41 kiloyears to 100 kiloyears during the mid-Pleistocene transition toward 1 million years ago. And also shown here are the timing of major tectonic events and oceanic events during the late Pliocene. However, preceding that colder Pleistocene was the prevalence of a relatively warm global climate during the earlier Pliocene epoch. And the causes and driving forces of this transition from a greenhouse Pliocene to an icehouse Pleistocene is still highly debated. And this just shows that we are still far from fully understanding the dynamics of the Pleistocene climate evolution. So aside from those oceanic events marked in the previous figure, paleorecords indicate a number of significant changes in climate patterns since the last 5 million years ago. And among this are the shoaling of the thermocline, cooling of coastal upwelling zones in the subtropics, cooling of the high latitude deep oceans, contraction of the tropical belt and oceanic warm pool, and emergence of strong temperature gradients along the equator. And all this together imply a large structural change in climate with major global and regional implications. So also along with the physical mechanisms happening in the ocean is the parallel change in the atmospheric condition. And this figure over here is a multi-proxy Pleistocene record of the atmospheric carbon dioxide evolution compared to the EPICA ice core record, atmospheric CO2 concentrations from boron isotopes of planktonic foraminifera, and alkanone-based PCO2 reconstruction from the ODP site 925. And this is in comparison to the present PCO2 record. And here we can see that the Pleistocene PCO2 values range from 250 to 300 ppm and are broadly comparable to the EPICA record. And I just want to show this figure to show you that PCO2 levels were higher during the Pliocene than during the Pleistocene. So the early Pliocene is the most recent period in history with a climate that is warmer than today. And this time interval includes two distinct periods of global warmth. One is during the early Pliocene or the early Pliocene climatic optimum from 4.4 to 4 million years ago of the Pliocene, which is the mid-Pliocene warm period from 3.3 to 3 million years ago. And these two intervals are considered possible analogs for future climate conditions because of the similarities of these records to the present atmospheric CO2 concentrations and global temperature records. And this figure here also shows a robust compilation of published analytical, instrumental, and modeled sea surface temperature records, shown here in y-axis, using a variety of geochemical proxies across all ocean basins and north-south and east-west comparisons over the last 5 million years. And this highlights the elevated temperature during the early Pliocene. And with that robust paleoceanographic reconstructions that have been done over the past several decades, this region over here is still relatively underrepresented, especially over deeper timescales. And the new sedimentary archives collected during the IODP Expedition 361 will fill this gap in the record with the availability of new high-resolution sediment materials from the Agulhas region in the southern Indian Ocean. So the Agulhas region is a key location for the global thermohaline circulation system because this area over here is where the Atlantic, Pacific, and Indian Ocean water masses interconnect, and it is therefore important for transmitting climate signals from one region to another. The Agulhas water mass is sourced from the Indonesian through flow, and it flows around the southern tip of Africa, where part of these waters leak or escape to the Atlantic through the shedding of these eddies over here, and it is termed the Agulhas leakage. And this leaked tropical waters then continue to the North Atlantic. The other component of the Agulhas current retroflects or returns back to the Indian Ocean, and this is known as the Agulhas return current. So the Agulhas return current is a strong current along the subtropical front over here, which is also called the subtropical convergence zone. And our study area sits within the framework of the Greater Agulhas Current System, located within the direct influence of the Southern Ocean frontal system, and with a recorded modern temperature of about 11 to 17 degrees Celsius. So the presence of these different frontal systems, namely the polar front, the subantarctic front, the subtropical front, contribute to the region's complex biogeochemistry with changes in phytoplankton distribution and variations in primary productivity. These are NASA satellite maps of modern chlorophyll concentrations showing the chlorophyll concentration, Coccolithophores, and diatom concentrations. And from this map, we can see a zonal band of high chlorophyll A concentration at the confluence between the subtropical front and the subantarctic front, this faint green over here. And during the winter, lesser chlorophyll concentration has been detected, and we can see here that the chlorophyll pigment is dispersed over here further up north. And in these two figures, we can see a distinct separation between the two phytoplankton groups during austral summer. So in the north, a Coccolithophore dominated community, and in the south, a diatom dominated phytoplankton community. And these two phytoplankton groups are controlled by the latitudinal position of the frontal system, specifically the subtropical front, with modern abundances that is driven by one or a combination of parameters such as nutrient availability, water column stratification, and changes in temperature. So in the fossil record, a previous study by Flores and others from the Cape Basin proposed that the subantarctic zone in the Atlantic sector of the Southern Ocean is mainly colonized by Coccolithophores instead of diatoms and other siliceous organisms, and that most of the export production over the last 500,000 years in this region was due entirely to Coccolithophores. Here in this top figure, studies show that the modern highest concentration of nutrients in the surface, and this is found in the upwelled waters located south of the polar front. And it is located specifically between the polar front and the sea ice. And this also consequently releases a significant flux of carbon dioxide to the atmosphere. And moderate Coccolithophore production has been recorded in the subantarctic zone. Here in the lower panel, the northward expansion of the sea ice during the last ice age produced limitations in surface ocean primary productivity south of the polar front. And this is what is being observed for the present day. So during this period, the westerly wind cells were displaced north of the polar front, and the main glacial production took place in the subantarctic zone. And at this latitude, this is also the same location as site U1475. And this phenomenon resulted in a net reduction in Coccolithophore production occurs during glacial period as a response to enhanced upwelling with the westerly wind cells being displaced further up north. And the glacial white ocean occurs both north and south of the polar front with two scenarios. So one scenario is due to the sea ice expansion in the subantarctic zone, and the other is due to the Coccolithophore blooms in the most temperate regions or the subantarctic. So in general, Coccolithophores are marine unicellular flagellate phytoplankton. They are considered one of the most abundant phytoplankton groups. Coccolithophores are sensitive to changes in water column characteristics, and their growth depends on the availability of sunlight and nutrients in the water column. They secrete calcareous outer coverings, and together with foraminifera and theropods, they are considered the ocean's most important suppliers of calcium carbonate, both in the seafloor and in the water column. So the fossil record of these organisms have been remarkably abundant and continuous since its first occurrence during the late Triassic, and constitutes a major part of deep sea sediments. So calcifying haptophytes such as Coccolithophores play a significant role in the global biogeochemical cycles, and these organisms have gained increased attention as they make vital contributions to oceanic primary productivity and play an important role in the carbon dioxide-oxygen exchange between the ocean and the atmosphere. In particular, Coccolithophores also may alter seawater characteristics through photosynthesis and calcification, and in turn, their distribution in the water column is controlled by seawater physical and chemical properties. So during photosynthesis, large amounts of carbon dioxide are sequestered by these organisms, and through calcification, the CO2 is released back to the atmosphere. And using sediment materials from the subtropical Indian Ocean, our study aims to address the following questions. The first question is, what are the drivers of Coccolithophore community change in the subtropical Indian Ocean during the early Pliocene? And in terms of Coccolithophore evolution, the Pliocene is regarded as an interval of relatively high evolutionary turnover. So the second question is, how did the evolutionary change in Coccolithophore species or species groups impact the global carbon cycle? So to address these questions, we have investigated sediment materials from site U1475 in the Agulhas Plateau in the Southern Ocean sector of the Indian Ocean across the early Pliocene warm period between 4.95 and 3.19 million years ago. And here we analyzed the samples for Coccolithophore assemblage composition and performed morphometric measurements using CALCITA software to present a comprehensive estimate of calcification using the measured thickness, mass, and volume of Coccolith calcite. So for the results, one of the striking findings of our study is the high percentages of reworked species at certain intervals. Because they are unusually that many in one field of view, I have counted the reworked species separately and also counted the rough numbers of diatoms in the same slides used for Coccolith analysis. So in here, we have separated the autochthonous nanofossils, or the total nanos here in blue, versus the alloctonous, or the reworked, in orange. And these reworked species are no longer included in all the succeeding calculations for the nanofossil fluxes. So the diatoms in yellow here is also very interesting, as it appears to be following the peaks in reworked at some intervals. So in this figure, we can see three prominent peaks. One is at 4.75 to 4.6 million years ago, before the early Pliocene climatic optimum. Some regular peaks during the early Pliocene climatic optimum over here, and another large peak at around 3.65 to 3.45 million years, and during the mid-Pliocene warm period from 3.3 to 3.19 million years ago. So reworking of coccoliths could indicate a number of potential mechanisms happening in the sediments or in the ocean floor where these sediments are deposited. This reworking could indicate, for instance, river fluxes, which we can already exclude, knowing that our study area is not within close proximity to a river. So another reason that we can look at is sea level change. So the drop in sea level exposes the continental shelf, which might erode during periods of increased humidity and could be carried to the study area by surface current. So here we have plotted the modeled sea level data, and it looks that some of the peaks and reworking can be explained by the changes in sea level. However, there are also peaks that do not go well with sea level change, like the biggest peak here during the early Pliocene. So high reworking percentages can also be explained by turbidite or contrite deposits, which are indicators of bottom current intensities. For example, in the Mediterranean, reworked species was used to monitor the exit of current from the Mediterranean and suggestive of an increase in the intensity of the bottom currents or the bottom current activity of the Mediterranean outflow. And this could also be considered as one of the potential mechanisms that can explain the high amounts of reworking, given that our study area is in the Agulhas Plateau and located on a sediment drift, and could further be suggestive of the intensity of the North Atlantic deep water exiting the South Atlantic going into the Indian Ocean. So in our study area, the interocean exchange of the North Atlantic deep water into the South Indian Ocean from the South Atlantic occurs mainly in the Agulhas Passage over here. However, there are also studies that propose that some of the North Atlantic deep water flow also extends around the southern flank of over here, the southern flank of the Agulhas Plateau, resulting in the deposition of a suite of conchorite sediment drifts. So the process is, winnowing or scouring of the ocean floor could bring high amounts of these reworked materials, and this process cleans or filters the smaller particles and leaving the big ones. And this is shown in the opposite trends between the total nanofossils and the reworked species. So most, or if not all, of the reworked species are considered big ones. So big ones which are greater than 10 microns compared to the autochthonous ones, which are as small as 2 microns. So these three prominent peaks, 2, 3, over here, could therefore mean a particular change in bottom current activity, or could be caused by changes in sea level during these times, or a combination of both. So for the actual results, without the reworked species, at site U1475, we have identified a total of 25 species and species groups comprising a mixture of taxa, typical of tropical to subtropical environments. So the assemblage is dominated by Reticulofenestra species with all the sizes together comprising relative abundances between 40 to 90% of the total Coccolithophore assemblages, all these yellow shades over here. So we have smaller than 3 microns, 3 to 5 microns, 5 to 7 microns, and greater than 7 microns. This abundance is followed by Coccolithos pelagicus and Gephyrochapsa species, both with relative abundances of up to 30%. And other species with consistent occurrences, but with very low contribution to the assemblage, are the deep photic taxon Florisphaera profunda, this blue over here, and the high nutrient indicator species Calcidiscus leptoporus. It's not plotted here, which both occurs in its large and small forms. So the small forms is associated with nutrient enhancement, and the larger forms, or the winter type, is indicative of cooler water conditions or high nutrients or both in the water column. So this assemblage composition at the Agulhas Plateau, which is in the subtropical region, is so much different from the other sites, like for example, at site U1476 in the equatorial Indian Ocean region. So here at site U1475, the very low abundance of Fluorisphaera profunda, comprising only 10% of the assemblage and almost no discoasters, which are species typical of oligotrophic or warm conditions, could indicate that the subtropical region was consistently bathed with surface waters with temperature that is so much lower than in the equatorial region, even during the early Pliocene warm period. And this is also supported by the high abundances of Reticulofenestra and Coccolithos pelagicus, which are both typical of high nutrient and cooler water conditions. So these are the absolute fluxes of the dominant species from the study area. These four plots over here are the Reticulofenestra, which is classified according to size fraction. Smaller than 3 microns, we have 3 to 5 microns, 5 to 7 microns, and greater than 7 microns. The second plots here are zoomed versions of these almost flat lines here, just to see the trends clearly. And in here, in the lower part, is the total abundance flux of all the Reticulofenestra size classes. All this just plotted together in one axis. So the abundance of this species, Reticulofenestra, indicates high productivity conditions. And in here, in the next figure, the total nanofossil flux is plotted in green. These two plots here are Coccolithos pelagicus and Coccolithos, which I also classified according to size. Pelagicus, which is smaller than 10 microns, and Brarudae, which are those that fall larger than 10 microns. And these species are indicators of high nutrient or cooler water conditions, And I also plotted here Fluorisphaera profunda. The abundance of this species in the water column reflects a deep nutricline or thermocline and a stratified water column. So I'll discuss these records according to specific time periods, and let's start with this area over here before the early Pliocene climatic optimum. So from the Coccolith record, before the early Pliocene climatic optimum, cooler water conditions occur at the study area, as shown by the high abundances of Coccolithos pelagicus and Coccolithos brarudae. And also, during this time, surface water is generally mixed, except for these two small peaks here at 4.8 and 4.7. But this could not necessarily mean upwelling, but continuous mixing of the upper 100 meter of the water column. And this mixing could have resulted in the increased abundances of both the smaller and the larger Reticulofenestra with increased productivity in the surface water. So in here is the carbonate record. And this shows that carbonates have highest percentages from 4.95, from this interval 4.95 to 4.4 million years ago, coinciding with the abundance of the larger Coccolithophores. All these Reticulofenestra greater than 7 microns and the Coccolithos, these are all large species. And also major contribution to this carbonate budget would be coming from the other sizes of Reticulofenestra, which all register highest abundances during this time period. And one of these productivity peaks coincide with high productivity conditions recorded in East Antarctica, plotted here in green, at 4.7 to 4.6 million years ago. So during the early Pliocene climatic optimum, this yellow area over here, the beginning of the EPCO is characterized by a slight increase in stratification, as shown here by Fluorosphera profunda. And there are also sharp decreases in the cooler water species Coccolithos pelagicus, as seen in here. And although this species has recovered at some point during the warm period when stratification relaxes. So here we see a decline in all Reticulofenestra species, with major contribution only coming from the smaller ones, which are the smaller than 3 microns and 3 to 5 micron size classes. So the decline happens at the beginning of this interval, but we can see here that an opposite increase in carbonates there's an opposite decline in carbonates with the increase in the Reticulofenestra. So this means that contribution from other calcifying organisms, such as foraminifera, could be a factor. And because this interval is also synchronous with the high productivity conditions in East Antarctica. So the reduction in carbonates during this time period from here is again due to the lesser contribution of the dominant species Reticulofenestra. So after the early Pliocene climatic optimum, this area over here, we can see that the major contribution to the carbonate budget was coming from the smaller Reticulofenestrids, or those that are up to 5 microns, recording highest peaks at 3.6 to 3.3 million years ago. And there are no significant contribution coming from the larger Reticulofenestra during the mid-Pliocene warm period, when a decline in the abundance of the smaller ones occur. So during this time here, stratification is stable with a small peak in Fluorosphera profunda from 4.4 to around 3.6, and then it's stable from 3.6 to around 3.1. So F profunda records show mixing from 3.6, which could explain the recorded increases in smaller Reticulofenestra and the Coccolithos pelagicus over here. So these are the results of the morphometric analysis of Reticulofenestra coccolith calcite of the different size classes, showing mass, thickness of the coccolith, which reflect the extent of calcification. And then the K value over here is the volume normalized measure of calcification and directly correlated to peak to POC ratio. These morphometric parameters are plotted against the calcium carbonate concentration from the sediments. A very low resolution PCO2 concentration calculated from alkenones in a study by Secchi. And this also shows an independent Coccolithophore signal. And then also plotted here is the LRO4 stack. So the general trend observed here in our record throughout the studied interval is increasing calcification, decline in calcium carbonate with decreasing PCO2. So during high carbon dioxide concentrations, Coccolithophores are not doing well because of factors such as increase in temperature and ocean acidification. And here we observe that there is an increasing degree in calcification, but this is for all the size classes combined. And also, ideally, if there's low calcification, this means that there's smaller species and thus smaller coccoliths. So it is assumed that smaller coccoliths will have lighter coccolith mass, and larger coccoliths will have heavier coccolith mass. However, results from our morphometric measurements of the Reticulofenestra size classes show that the relationship between size and coccolith mass is not always perfectly correlated. So here we show a decreasing correlation decreasing correlation between the size classes and the coccolith mass. So for instance, the smallest Reticulofenestra, or those smaller than 3 microns, has a correlation coefficient of 78%. The coefficient decreases for the 3 to 5 microns, which is 70%, and even lower for the size classes 5 to 7 microns, which is 50%. So this means that the smaller size classes have uniform morphometry. And this is in contrast to the large size classes, which have more diverse species, as shown by different shapes of larger coccoliths, some of them becoming more elliptical or circular at certain intervals. And also, the central area of these species are sometimes filled with calcite or has an open central area. So during the early Pliocene climatic optimum, our data show a decline in calcification degree with increased carbon dioxide concentration. And previous studies suggested that atmospheric carbon dioxide is considered an important driver of Coccolithophore evolution during the Cenozoic. So the decrease in atmospheric carbon dioxide would reduce the aqueous concentration in the surface oceans, and this leads to a limited supply of carbon for the Coccolithophores to grow. So one of the adapting mechanisms of Coccolithophores is to produce smaller coccoliths, and this has been shown in older time intervals where a reduction in cell size was found across the Eocene-Oligocene transition during a dramatic decline in atmospheric carbon dioxide drawdown. And this has also been documented in different ocean basins from the Miocene to the Pleistocene. So in conclusion, sea level change brought reworked materials from the shelf. 3 distinct peaks in reworking may indicate bottom current activity or intensity. The low abundances of Fluorosphaera profunda and absence of discoasters that are typical of oligotrophic and warm conditions, and the high abundances of Reticulofenestra and Coccolithos pelagicus, which are species typical of high nutrient and cooler water conditions, could indicate prevalence of lower temperature over the APT during the early Pliocene. So the lesser abundance of Fluorosphaera profunda, comprising below 10%, also indicates a mixed upper ocean all throughout the studied interval, although this could not be considered upwelling, but there is no strong upper ocean stratification. And this is for the upper 150 meter of the water column. That lith size and calcification is influenced by PCO2 during the early Pliocene climatic optimum, with high CO2, smaller liths, and then reduced calcification. However, on other intervals, regional factors such as nutricline depth, stratification, and temperatures drove a Coccolithophore community change more than the concentration of CO2 in the atmosphere. And the overall trend toward the small coccoliths, from larger ones to small coccoliths through the Pliocene to the Quaternary, has been previously documented in a number of studies, and this has been linked to species turnover and extinction events. And this shift in the production of coccoliths from larger to smaller ones may have played a key role in the later decline of PCO2 and cooling during the Pleistocene. So with that, thank you very much, and I would be happy to take any questions.
Okay, thanks very much for that interesting talk. I don't know if we've had any questions in yet, so make sure you type them into the Q&A box if you have any, and I'll pass over to Aidan.
Yeah, thank you. Thank you very much for that, Debs. That was a super interesting talk. While we wait for some questions to come in, I had a couple of questions myself. The first of which, I wondered how straightforward it is, or whether there's a link between the mixing and the stratification of the upper water column and the position of